case you missed, right? Okay, good morning once again, boys. If you recollect from your memory, we were talking of the new chapter, that is molecular basis of inheritance. And this chapter, as I reminded you time and again, is not only important just for the sake of your CBSE board exam or your NEET exam or UPSC ND exam. It is very, very useful in our general day-to-day life. Unless and until you understand why you are what you are, it is very difficult to proceed in our life. So basically, you need to understand that whatever you are today, you are 6 feet height, you are 1 ton weight, you are super intelligent, you are champion, or you are stupid, all this is because of our DNA. So basically I said DNA. Are you able to hear?
Yes, sir.
Yeah, good. So DNA is deoxyribonucleic acid. So expansion of DNA is deoxyribonucleic acid. Now what is it made up of? Where is it present? How is that it is responsible for what we are today and what we are going to be tomorrow? For that, we need to understand its anatomy. What is inside it? We need to understand the location. Where is it present? Since we call it in general nucleic acid, if someone asks you what is DNA, your answer is in a low level, low category, DNA is a nucleic acid. What do you mean by this? Nucleic acid. First word nucleic says DNA is present inside the nucleus. Acid because it is acidic in nature. So nucleic acid is something that is acidic in nature and being present in the nucleus. Is it alone? No. Along with it, there is one more nucleic acid called RNA, Ribollose nucleic acid. Why are we saying that deoxyribulose and ribulose in DNA and RNA? As I told you, one atom of oxygen is less in DNA. It is removed. So only we are using the word deoxy. D means minus, minus of oxygen. Hence, in ribulose, what is ribulose? You may recollect, ribulose is a sugar, like glucose is a sugar, fructose is a sugar, lactose is a sugar, maltose is a sugar. If you see, like Rakesh, if you write in English, spellings OSC, OSC, maltose ending with OSC, sucrose ending with OSC, glucose ending with OSC. Anything that ends with OSC is a sugar molecule. Since ribulose also ends with OSC, ribulose is a sugar molecule. So, in sugar molecule, ribulose sugar molecule, one atom of oxygen is removed. Hence, deoxyribulose. I think I have shown you the structure of sugar molecule. Where did I show? Yes, here is that. Look at that. Okay. So, that is pentose sugar. The other day I said. Penta, 5. Okay. So, 1, 2, 3, 4, 5. 5 carbon atoms are there. So, we say it is a pentose sugar. And one oxygen is less. Hence, deoxyribulose. Whereas in ribulose, all are present, including oxygen. Hence, we say ribulose. So, the DNA is made up of first molecule, sugar molecule. What type of molecule it is? Pentose sugar molecule. Clear? This is how the structure. The second molecule that will be present in that is phosphate group, phosphorus. Okay? And the third one that is present is nitrogenous bases. So, DNA or RNA in general, nucleic acids are made up of three chemical compounds. One, sugar molecule. Second, phosphate molecule. Third, nitrogenous base. And I said, third one, nitrogenous bases, depending upon their structure, they are of two types: purines, pyrimidines. In purines and pyrimidines, just by seeing the structure only you understand. Purines are double ringed structure. Two rings are there. Look at that. Whereas pyrimidines, only one ring is there. Single ringed structure. Let me expand that. Yes. As you are seeing here. In double ringed structures, purines, there are two, adenine and guanine. So how to remember in the exam, purines? Take the first letter of each. Rajwal is waiting. Okay? Right. Let me see whether it is. Yes. Okay. So the first letter of each, if you pick up, purines P, adenine A, guanine G. See, if you take reverse, GAP, gap. This is how you should remember. You, for everything you just put a abbreviation or a code word, easy to remember in the exam. So GAP. Okay, GAP. Why Prasad is not able to hear, it seems. I have put full volume. Okay. Ask Prasad to adjust his volume. Rakesh, are you able to hear? Hello?
Yes, sir, audible.
Why only Prasad has got, maybe he has kept the volume. Yes. So, the abbreviation for purines, you should remember it as GAP, G-A-P. G for guanine, A for adenine, and these two are purines. Okay? Yes. PAG. And the second one is pyrimidine. And how many are there? Three are there: cytosine, thymine, uracil. Okay? So here PCT, pyrimidines, or PCTU, because you may get confused. Purines also first letter P and pyrimidines also first letter P. So for purines you use just one letter P. Pyrimidines you use PY. So in pyrimidines PY, cytosine C, thymine T, and uracil U. So if you join this, PCTU or PIC2, PIC2, PYC, TU. So that easily you can remember in the exam. Sometimes you will write ulta. What are purines? You will write cytosine, thymine, uracil. What are pyrimidines? Rakesh will write adenine and guanine. So don't get confused. Purines are GAP, GAP, GAP. Guanine, adenine, purine. And structurally they are double ringed structures, as you are seeing. And PIC2, PIC, PY, pyrimidine, C, cytosine, T, thymine, U, uracil. So PIC2. So here, structurally they are, as you are all seeing, single ringed structure. Okay? And where is this uracil present? Uracil is present only in RNA. So this is one of the basic differences between DNA and RNA. Already I have told one difference. What is that? Ribollose, deoxyribulose. In DNA, deoxyribulose is the sugar, pentose sugar present. In RNA, full-fledged ribulose sugar is present. That's one difference. Second, in RNA, uracil is the pyrimidine present in place of thymine. Whereas in DNA, it is thymine that is present. So two differences. And third difference, already by just seeing the diagram only you understood. What is that? The very first diagram. Look at that. DNA is a double thread. That's why, if you recollect from your memory, Watson and Crick said double helix. Nichenege, I said. If you twist the Nichenege ladder, how it will be? That's how DNA. And RNA is single thread. And that single thread is twisted, as you are seeing in the photo. So these are the three differences you can always speak of between DNA and RNA. And another difference already we said, that DNA, RNA both are present in almost all the organisms. But in viruses, only RNA is present. So 4 differences you can write. And how they are interconnected. Now these 3 molecules, which is that pentose sugar, one nitrogen base, one phosphate molecule. These 3 are interconnected like this. Then such a structure is called nucleotide. Nucleotide. Okay? What is a nucleotide? It is the portion of DNA. What type of portion it is? A portion that contains all the 3 chemical compounds together. What are those 3 chemical compounds? Sugar molecule, phosphate molecule, one nitrogen base. So that is a nucleotide. One nucleotide is attached to another nucleotide with the sugar and phosphate. So look at the giant, sugar and phosphate. Since one nucleotide is joining to nucleotide 2, nucleotide 3, nucleotide 4, like this, it is easy now for all of us to define DNA. What is a DNA? DNA is a polynucleotidal chain. Then you add whatever we said in the beginning. DNA is a polynucleotidal chain having acidic nature present in the nucleus of a cell. That is the complete definition of this. Okay? And what? Yeah, it is acidic in nature. Okay? So, DNA is a polynucleotidal chain. In turn, what is nucleotide? If you take one monomer, nucleotide is a monomer. When all the nucleotides, many nucleotides join together, as our Sandeep knows, it is a polymer. Monomers join together to form polymer. So we say DNA is a polymer. What is the monomer of it? Nucleotide. What is nucleotide? Sugar molecule, phosphate molecule, nitrogen base. If these 3 join together, that is a nucleotide. Since one nucleotide joins with another nucleotide, it is called polynucleotidal chain. Okay? I think all this we have covered. This also we have covered. Pyrimidines. And this is how the DNA look like. Double helix. The ladder is twisted. And in each, and there is a twisted structure like this, how they are joined together? A nitrogenous base, we said that there are 2 nitrogen bases, purine and pyrimidine. That either purine or pyrimidine is joining to pentose sugar. And that bond, linking nitrogenous base with sugar is called glycosidic linkage. And when these 2 molecules, nitrogen base and sugar, okay, S, sugar, N, nitrogen base, is equal to NS. NS means nucleoside. So this is how you must remember in the exam. What is a nucleotide? What is a nucleoside? Or you will ask you 1 mark. Differentiate between nucleoside and a nucleotide. Give an example, then you have to do this. So how to remember first without confusion? SN is equal to NS. Or even in nucleoside spelling only, first one is N for nitrogen base, you remember. And go for the spelling full, Nucleos. S for sugar. So in the word itself, answer is there. Nucleoside. N for nitrogen base. S for sugar. The question is only giving the answer to you. So what is a nucleoside? You know it. N for nitrogen base. S for sugar. Then he is asking differentiate nucleoside with nucleotide. Add one phosphate, because you know after all nucleotide is made up of 3 substances. 2 are already there, which are called nucleoside. So nucleoside plus phosphate molecule, nucleotide. Easy, isn't it? This is how you must have abbreviations so that you can remember easily in the examination. We said polynucleotidal chain. So specific names are there depending upon the nitrogenous base. Adenosine, in the case of RNA, the nucleoside is adenosine because nitrogen base is adenine and sugar molecule. So adenosine. Guanine plus sugar molecule, guanosine. Cytidine plus sugar molecule, cytidinase. Uridine, uracil plus nitrogenous base. In case of DNA, deoxy will add deoxyadenosine, deoxyguanosine, deoxycytidine, deoxythymidine, and already I think some of you are very good boys. You have drawn all these diagrams. You have practiced. Okay. Those who are still not done, you must do. Now, this is a nucleotide. And what is important here, you must understand, is the position of the carbon atom. How to give carbon atom its numbers, already Desai sir must have covered in organic chemistry. Benzene, toluene, C6H6 when you write, how to give the number. So see here in the sugar molecule, pentose sugar molecule, carbon number 1, 2, 3, 4, 5. And where is this nitrogen base attached? Carbon 1. Where is phosphate attached? Carbon 5. So we say 5 carbon. Here we say 1 carbon. Right? This is how you must write a nucleotide structure. So as is giving you nitrogen base plus sugar plus phosphate is equal to nucleotide. So deoxyribonucleotide. To be specific, how will you define DNA now? It is a polymer of deoxyribonucleotidal chain. Am I correct, Kadakul? Yes. Now, as we said, it is the bond. Which is the bond that is connecting each other? We said just now glycosidic bond. Here there is another bond, phosphodiester bond. Again, you must have heard in chemistry, esters, esterification. Here it is diester, 2 esters. Who is attaching it? It is the phosphate molecule. Look at the diagram. In the dinucleotide, one nucleotide is attached to another nucleotide through this phosphodiester bond. Luckily, as you go through this chapter, you will understand that the beauty of the God's creation. We say somebody from IIT engineer and somebody from NIT, Surathkal engineer, all that. But in front of God, you will appreciate these are nothing, whether IIT or even Massachusetts Institute of Technology, isn't it? And these bonds are created in some places very strongly. So it is very difficult to break that. But in some places, God's creation is such that just by applying a little bit of amount of energy, the bond breaks. And that creates varieties. See, all of us, if you take our own 135 crores we are, Indian population, isn't it? But no two individuals are same. We are all different, though we are all basically human beings. And that is because of this bonds where... Yeah.
Constituent is concerned, but arrangement is different.
Ah, exactly. And how that arrangement will be different? It is because of this loose bonds. Varieties can be created. And we human beings, being most intelligent among all organisms, somebody like Kadakol intentionally breaks that. He will do some kitapati. He will change that and create new sequence of genes. When new sequence of genes are arranged, varieties are formed. That means Kadkol is challenging the God. What is that? God, if you can create me, I can create something else. But question mark is there for Kadkol. What is that? God's creation, all are good boys and good girls. Basically, God creates all of us with a good nature. But how we become during our lifespan will be decided by the environment in which we are living. Okay? You must have heard that story, A parrot is grown by a Purohit or a Swami. And the moment Kadkol goes there, what does he say? The moment Kadkol goes to that ashram, hello, good morning Kadkol, please aaiye, baithiye. Okay? Language is sophisticated. But if the same parrot is taken to a rowdy, a gunda shita, how does it speak? The moment Kadkol goes, it starts abusive language, foul language. Parrot is same, genes are same, but the environment in which the parrot was living was extremely good with Swami. Environment in which presently it is shifted, bad. So only your parents will be telling you, select good friends. Your friends should be good. And many times the psychologists say, tell your 5 friends names, I will tell you what you are. Okay? So only you must be very careful. Your parents also will tell, hey, be careful, avun jote hogbeda, ivun jote hogbeda, isn't it? Why? This is the thing. Your environment influences. That's why we are the products of not just our DNA, Kadkol. We are products of our DNA as well as our environment. Okay? That's why in your health education you got a chapter, Heredity and environment. It's not just heredity that will decide. It's the environment. And for all this, whatever we are speaking today, thanks to this gentleman, look at that. Aisha and Professor Watson and Crick, all our favorite, and Wilkinson and Franklin. Without these people, we wouldn't have understood what we are, why we are. Thanks to them. What is that they have said? Aisha, way back in 1869, identified DNA and named it as nuclein. You must appreciate him. When? 1869. Now we are 2021. We are all boasting of ourselves with electron microscope, nanotechnology, what else could call? Stem cell technology. But look at that, 1869. That's how you should appreciate, Rakesh. What about Professor Watson and Professor Crick? They are responsible for this double helix model because until 1963 we were not knowing where is this DNA, what it is, where it is, nothing. So thanks to them, and suitably they were awarded Nobel Prize, thanks to the people who are encouraging science worldwide. For everything, Kadkol, there should be encouragement. When I encourage Rakesh, Rakesh will do with full josh, and the result will be double. That is the reason why I always tell you, do something great and get appreciation. Okay? And third one, we said Franklin and Wilkinson's, of course, X-ray diffraction. Whatever we are, even today, we are using all the technology of X-rays. And what is the distance? Some of them, they brought mathematics into our biology. They started measuring what is the distance of the DNA, total length of DNA, how many twists are there, and what is that helical structure is made up of, all that. So some of the mathematics is DNA is made up of two polynucleotidal chains coiled in right hand fashion. Okay. Why two polynucleotidal chain? One nucleotide chain made up of sugar, phosphate, nitrogen. That is one. Let me show you. Prajwal has left the meeting. Okay. So look at this. You will understand. Okay. Now I think you will understand better. Look at that. This is how we are calling DNA double helix. So first I said Nichenige ladder. Look at that. So you have got one group, what I say, I can use the word vertical thread. Vertical, male in the third, vertical head. So two vertical threads are there, see. One we are naming it as the 3' and 5'. Another one we are putting it as 5' and 3'. What is this 3' and 5'? Carbon number attachment. Okay. And in the ladder, how you have got steps. Like that, here also you have got steps. What are those steps made up of? Base pairs. What base pair? Nitrogenous base pair. And I said base pair ruling. Adenine, or in general I'll say, one purine combines with another pyrimidine. And reverse also is applicable. One pyrimidine combines with purine. So adenine always pairs with thymine. Guanine pairs with cytosine. And reverse also is there. Cytosine pairs always with guanine. Thymine pairs with adenine. Why I am saying this? Do not entertain yourself that there will be pairing within adenines, within purines or within pyrimidines. It is always purine versus pyrimidine. Okay? And look at the base pairs where they are present. And look at the thread, vertical thread. And I said again, in the examination it is easy to remember. The vertical threads are made up of SP, superpentapolase, sugar plus phosphate. So vertical threads are made up of SP, sugar plus phosphate. What about the central steps? They are made up of nitrogenous bases. I hope you understood this base pair ruling, the total structure, and one side Kadkol will hold, other side Prasad will hold, and twist it towards the right. So right direction orientation. Then naturally when Prasad twists this, you get loops or grooves. Okay? And somebody, as I said, measure the length. And these are the measurements. What is that? How much? DNA is made up of two polynucleotidal chains coiled in right hand fashion. You understood that? Pitch of the helix is 3.4 nanometers or 34 angstrom units. Number of base pairs in each turn 10. And the distance between adjacent base pairs 0.34 nanometers or 3.4 angstrom units. So backbone of DNA is formed.
Can ask these measurements.
No, nobody will ask. This is just to know, so that you have an idea what would be the length of DNA in us, total length. It is only for appreciation, that's all. So backbone of DNA is formed of sugar and phosphates. The bases project inside steps. Two chains have anti-parallel polarity, just now I said. One is starting with 5 carbon attachment, ending with 3 carbon. Another starting with 3 carbon, ending with 5 carbon. So we say 5' to 3', another one 3' to 5', just now I said. Look at that. Yes. I hope all of you are able to see this. Top 3', down 5'. Here, right side, top 5', down 3'. This is what we call anti-parallel. They look like railway lines, you see. Railway track will be there, no? Like that. But property wise, they are 3 prime to 5 prime, another one 5 prime to 3 prime. This you must remember. Anti-parallel. Though structurally they are parallel, but polarity wise they are anti-parallel. So that is the structure of DNA. And now further you can see the base pairs. The base in two strands, of course you know, is connected with hydrogen bonds. Again, hydrogen bond is weak bond, as all of you know. And the base pair is abbreviated as BP. And how these are attached? Nitrogen bases are attached with specific bonding, number of bonding, and the bonding is hydrogen bonding. One is A double bond T. Now look at that. This is also you must know. A adenine combines with thymine with double bond, whereas cytosine combines with guanine with triple bond. So between A and T, 2 hydrogen bonds are there. Between C and G, 3 hydrogen bonds are there. So pyrions comes opposite to a pyrimidine. This generates uniform distance between the 2 strands. Okay. Now look at this. We'll see the diagram. Yes. Now look at that. I hope you understood here. This is the connection. Yes. Look at that. Base pair. On left side thread, if you see vertical one, top phosphate molecule is there. Down OH molecule, hydroxyl. And where is OH in the right side thread? Top OH. Down phosphate. This is anti-parallel. And see the bond. There are 3 bonds. One is sugar molecule attaching to nitrogenous base. One bond is there. I hope you are able to understand. Another one is sugar molecule with phosphate. See the bond between 2 sugar molecules in the same ladder. See only one ladder, vertical one. You can see here. So there are 2. And third bond is there. What is the third bond? Hydrogen bond. Where is it present? It is present in between 2 nitrogenous bases. So totally there are 3 bonds. What is that, Rakesh? Are they James bonds? I think Rakesh has left. No? Must be sleeping?
No sir, phone call आ गया sir.
Okay. So, Rakesh, 3 bonds are there. Bond number 1.
Yes sir.
In between 2 nitrogenous bases, hydrogen bond. Between adenine and thymine, 2 hydrogen bonds. Between cytosine and guanine, 3 hydrogen bonds. Okay, that is 1 hydrogen bond. Second, between sugar and nitrogen, there is a bond, you see. Sugar and nitrogen. That black circle, encircled, I have encircled. Within that you focus. One is the hydrogen bond between cytosine and guanine. And second one, between cytosine and sugar molecule. What is that bond called?
Glycosidic.
Yes, good boy. Glycosidic bond. And the third bond, sugar and phosphate. Yes, Karkol? Phosphodiester bond. So totally in a DNA molecule there are 3 bonds. And which one among them easily can be broken? Which is the weakest bond out of the 3? With your chemistry knowledge, you tell me, Sandeep. Hydrogen. Yes. ನಮ್ಮ ಸಂದೀಪ್ ಏನು ಮಾಡ್ತಾನೆ, ಸ್ವಲ್ಪ ಬೆಂಕಿ ಹಚ್ತಾನೆ ಅಷ್ಟೇ. The moment he puts some energy, supplies some energy, hydrogen bond is easily broken. When hydrogen bond is broken, nitrogenous pairing is broken. That means DNA is broken. Sandeep calls it DNA replication. This is how DNA replication takes place. And what is the faida for us? What is the use of it for us? When DNA undergoes replication, Sandeep can play with DNA. I am using deliberately word play. He can create anything he wants. But there is a danger. Somebody like me will give precaution to Sandeep. Sandeep, jasti aata adbeda. Why Sandeep? Yeah, you are going to play and create something unknown. You don't know what is that organism. It may eat up Sandeep only. So only we biologists will be taking maximum precautions when we are playing with DNA. So in my next class I will tell you what are all that we can do because of DNA replication. Right? Until then, take care. Bye.